Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology boys, and this is for 15th October 2020. We are into the revision mode, as all of you know, and we are revising an all important chapter called molecular basis of inheritance. In that we were studying about the secrets of the characters that we attain, and in the process we studied about the DNA, its structure, RNA, types of RNAs, and an important thing called central dogma, wherein transcription, the conversion of DNA into mRNA. How exactly it takes place we have studied in our previous sessions. Now we are going ahead with genetic code. What is a genetic code? The genetic representation of codon by which the information in RNA is decoded in a polypeptide chain is called genetic code. So there is code, that is the code will be, or codon will be having 3 nitrogenous bases, and that 3 nitrogenous bases will be responsible for formation of linking of various amino acids so that proteins are formed. So it is the relationship of amino acid sequences in a polypeptide chain and the base sequences of DNA. When we say base sequences, it is nitrogen bases. So genetic code is the relationship of amino acid sequence, that you must remember, and that forms a polypeptide chain, and that polypeptide chain of amino acids will become protein, and this formation of protein is linked to the base sequences in mRNA. So that is genetic code. It directs the sequence of amino acids during the synthesis of proteins. So George Gamow suggested that genetic code should be combination of 3 nucleotides to code 20 amino acids. Like in our own H.G. Khurana, we are proud that he is from India and he has done a lot of work which the whole world appreciates. So H.G. Khurana developed chemical method to synthesize RNA molecules with a defined combination of bases. He has a lot of patience. He has worked out all the various combinations of nitrogen bases which combine together form a protein. So which are the nitrogenous bases? 3 nitrogen triplet code we say is responsible for the attachment of amino acids so that it will form protein. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. So what are the salient features of genetic code? As I said just now, the code is triplet. 3 nitrogen bases will be there. 61 codons code for amino acids and 3 codons do not code for any amino acids. Those 3 are UAG, UAA and UGA. So these are called stop codons. The process of protein synthesis will be stopped. Since they are not coding, we can also call them as nonsense codons. So 61 sense codons plus 3 nonsense codons will be there. And codon is unambiguous. Ambiguous means there is some confusion. Unambiguous, very clear, and they are specific. Code for a particular amino acids. A triplet base, nitrogenous base codes for one particular amino acid. The code is degenerate. The code is degenerates, means for a particular amino acid more than one codon can be used. This is called degeneracy of the code. That is, more than one codon. Remember this, more than one codon can be coded. The codon is read in mRNA in a contiguous fashion without any punctuation. It is comma-less, we say. It's a comma-less. The codon is nearly universal, irrespective of whether human beings or animals or plants, codon is universal. AUG has dual function. As all of you know, AUG is the initiating codon. It codes for methionine and also acts as initiator codon. So you remembered three just now. Terminating codons are nonsense codons. But the protein synthesis process starts with initiating codon AUG. How to remember? AUG, August. Remember, August is the initiating. And 15th August we got freedom. So the free India started like that. You remember, here also AUG has started the protein synthesis process. So it is a initiating codon. And it codes for which amino acid? MAT, methionine. And mutations and genetic code, a change of single base pair. Whenever there is a change in single base pair, we call it as point mutation. In the 6th position of beta globin chain of hemoglobin Hg results due to the change of amino acid residue glutamate to valine. This already we discussed. These results into disease called sickle cell anemia. Previous chapter we discussed it. Insertion and deletion of 3 or its multiple bases. Insert or delete one or multiple codons, hence one or more amino acids, and reading frame remain unaltered from that point onwards. Such mutations are called frame shift insertion or deletion mutations. So point mutation and base mutation. Then coming ahead, there is something called Weibull hypothesis. I think it's not mentioned here. You must remember. Weibull hypothesis was given by Professor Crick in 1965. According to this, third nitrogen base of a codon, for example, we take the initiating AUG. So 3 nitrogen bases are there. This is a triplet code we say. And according to Professor Crick, third nitrogen base of a codon binds loosely. That means this is the third one. It binds loosely with anti-codon, and codon is mainly specified by first 2 bases, these 2. Hence, the same tRNA can recognize more than one codons differing only at third position. This is called Weibull hypothesis. So, to brief, genetic code normally it's a 3-mark question. The characteristics of the genetic code he will ask you. So quickly you revise it. Genetic code is universal. It's found in all living organisms. So remember, universal. So genetic code is universal. And then for a particular amino acid, more than one codon can be used. This is called degeneracy of the codon. So you remember DG, degeneracy of the codon. And then a particular codon will always code for same amino acid. So code is non-ambiguous, non-ambiguous. But GUG, when present in beginning, codes for methionine, and when present in intermediate position, codes for valine. There's only one. And the codon which initiates the protein synthesis is called initiating codon, and the initiating codon is August, and it codes for MET. Codons which do not code for any amino acid are called nonsense codons, or stop codons, or terminator codons, and these are UAG, UAA, you can remember UAG and UAA, these two are common, first 2 nitrogen bases. And the third one is reverse, UGA. So UAG, UGA, UAA, these are the terminating codons or nonsense codons. And now we go to the tRNA, transport RNA. The tRNA has got a specific shape. It's a clove model, we say, lavanga, clove. The tRNA is called as adapter molecule. It has an anti-codon loop, this one, anti-codon loop, that has bases complementary codon present on mRNA. So on mRNA you got nitrogenous bases. These are called codons. On tRNA, at the loop, there is nitrogen bases, and these are called anticodons. So if there is a matching between codon and anticodon, that means it has to be sensible codon. Then the particular amino acid will be present on the or attached to the tRNA. And as the mRNA and tRNA move, so with the help of rRNA, of course all this thing will take place in rRNA, ribosomal RNA. One amino acid gets to attach it to another amino acid, like this chain of amino acids, and that forms a protein. So as like a railway bogie on railway platform, how the train moves, like that when the tRNAs move on mRNA is a railway platform and tRNAs are railway bogies, and rRNA is the railway station. As the bogies move, you can see one after the other amino acids get attached, just like the people who are traveling, they get down one after the other, like that they form a mob. Similarly here the mob is the protein. So tRNA plays a very important role in this protein synthesis. What is that it has got? It has got an anticodon loop, that's the main thing, and that has bases complementary to code present on mRNA, and also as an amino acid acceptor. This is the amino acid acceptor to which amino acid binds. tRNA is specific for each amino acid. The secondary structure of tRNA is depicted as clover leaf. Just now I said clove, lavanga. In actual structure, the tRNA is a compact molecule which look like inverted L. Okay, inverted L. And further, we go ahead with the background that for protein synthesis, first of all, the process of protein synthesis from mRNA is called translation. That you must remember. I said for protein synthesis, two steps are must. One is transcription, that is DNA to mRNA. Second is translation, mRNA to protein synthesis. So translation is the process of polymerization of amino acids. Many amino acids, 1, 2, 3, okay, to form a polypeptide, and that polypeptide is protein. The order and sequence of amino acids are defined by the sequence of bases in mRNA. Who will decide the sequence of amino acids that are formed? These codons. The codons present in the mRNA will decide the sequence. Amino acids are joined by, of course, peptide bond. So it involved the following steps. First, charging of tRNA, number 1. Second, formation of peptide bonds between two charged tRNAs. So the start codon is always August, I said. An mRNA has some additional sequence that are not translated, called untranslated region, UTRs. Okay, untranslated regions. For initiation, ribosome, as I said just now, this is ribosome, ribosomal RNA, rRNA. You got rRNA, you got tRNA, you got mRNA. All the three are involved in the protein synthesis, remember this. And on mRNA, you can see here triplet code. So there is 3 nitrogen bases together, and that's called codon. And this codon should match with the anticodons of tRNA. Once this matching takes place, what happens? The amino acid acceptor will be there. So immediately the amino acid will come and sit here. And next, as like a train, I said all this is happening in the ribosome. So ribosome, as you know, has got 2 units, one bigger unit and one smaller unit. Like if you recollect, 80S and 20S, or 70S and 30S, like that. So the entire process takes place in the ribosome, and as just like a train running on a railway platform, as the tRNA rRNA complex moves, the amino acids, depending upon the matching of codon of mRNA and anticodon of tRNA, amino acids keep on linking with each other, and that forms a protein. Okay, So basically this is what happens. So for this, the start codon must be always AUG, and some areas will be there which will not translate, that's called UTR. For initiation, ribosome binds to mRNA at the start codon. So ribosome is rRNA is binding it. Ribosomes moves from codon to codon along mRNA for elongation of protein chain. At the end, release factors binds to the stop codon, terminating the translation and release of polypeptide from ribosome. As the train moves, sometimes it gets a signal, red signal, it stops. Similar is the way. As it moves, the what we call nonsense codons or terminator codons will come. So once the terminator codons come on mRNA, the codon and anticodon will not match. That's why they are called nonsense codons. Once nonsense codons do not match with the anticodons, there will be termination. The amino acid chain gets cut off and the protein is released. This is what happens. So translation is the mechanism by which a triplet base sequence of an mRNA guides the linking of a specific sequence of amino acids to form a protein. Protein synthesis occurs over the ribosomes. Each ribosome has two unequal parts, as I said. One is like, for example, 70 years, another is 30 years or 20 years, like that. Okay, Steinbergs. And the larger subunit of ribosomes has a groove for pushing out the newly formed polypeptide and protecting the same from cellular enzymes. The smaller unit fits over the larger one like a cap but leaves a tunnel for mRNA. As you are seeing, the tunnel is there. The two subunits come together only at the time of protein formation. The phenomena is called association. Mg2 plus is essential for it. Ribosomes usually form helical groups during active protein synthesis known as polyribosomes or polysomes. There are three reactive sites in a ribosome. P site, that is peptidyl transfer or donor site. A site, amino acyl or acceptor site, and E site, exit sites. So P site, A site and E site, exit site. P site is jointly contributed by both ribosomal subunits. A site and E site are largely confined to the larger subunit of ribosome. So this is what happens in protein synthesis. And we continue with this, the regulation of gene expression, in our next class. Until then, take care, keep on practicing.